Fishes, there are more than about 37,000 described species of fishes on the planet. We're describing about one a day every year and have been for decades, so that number is continuing to increase. When you add up all of the other vertebrate groups, the amphibians, the reptiles, the birds, and the mammals, those all together don't sum to 37,000. So the fish diversity in vertebrates, fish win, absolutely, hands down.
I'm Laura Riley, and today we're diving into the world of fish, frogs, and reptiles, because sometimes the creatures slithering under logs or swimming beneath the surface are the best indicators of how healthy our planet is. Our guest today is Dr. Casey Dillman, curator of fishes, amphibians, and reptiles at the Cornell Museum of Vertebrates. He studies biodiversity, conservation, and the incredible and sometimes overlooked ways these animals can tell us what's happening in our ecosystems. We'll talk about what happens when amphibians vanish, why studying reptiles actually helps humans, and what a jar of preserved fish can tell us about climate change. Welcome.
Thank you.
It's
a pleasure
great
to be
meet
here.
you.
Yeah, you too.
So I did a little bit of homework, and it says that in your collection, which is at the Lab of Ornithology for all of you, you have between 1.25 and 1.3 million individual fishes in jars.
That's correct. We did the math recently. Everything's digitized.
So someone counted all of these.
We have cataloged everything. It's searchable, and there's over 1.3 million individual fish in the collection.
That is tremendous.
And about 1.5 million total specimens across all 4 vertebrate collections.
Wow. So I've heard you say that these are that there's kind of like a time machine element of this.
Yep.
How do you describe what you get from all of these jars of fish specimens?
That's a great question. It really depends on the question you're asking. Historically, specimens were collected for like biodiversity studies, like describing new species, trying to understand the planet, what lives where, and where it's distributed, how things are related. We're still doing a lot of that in fishes. There's about 1 species described every day, every year. More than like 350 species annually are described and have been for decades now. But we also are starting to use the collection with new technologies in different ways, because the organism comes from the environment, and so is a record of the environment that it came from. And we can look at what it was eating by doing dissections. We can also use mass specs or GC machines to look at stable isotopes, so we can see what something was, where it was in the food chain.
Can you say what is a mass spec?
Sorry,
just for a lay audience.
It's an instrument where you basically turn a piece of tissue into a gas, and you run it through a machine, and it can tell you the carbon and the nitrogen and the different elements that are in there. And that gives you a bit of information about what it was doing in the environment when it was collected, where it was in the food chain, and how it was getting nutrients moving up the food chain.
So I'm assuming that was one of the new technologies you were just talking about. What are some of the other new technologies that you're bringing to bear on this collection?
We do a lot of CT scanning with my group right now. Like we're actually taking all these fish from the collection, and we are scanning them in 3 dimensions to make them sort of models of what they look like inside. And it's really fun because the specimens themselves are very captivating, but sometimes we work with these really small things.
All right, you're going to have to convince us.
Okay.
Captivating. I'm imagining like a piece of fish in a jar.
It's a whole fish.
Tell us more.
The whole fish.
Oh, it's the whole fish.
The whole fish.
All of these is okay.
Yes, so 1.3 million whole fish. Well, we do occasionally take tissues from specimens now for genomic analysis, for sequencing DNA. But the organism itself has this sort of charisma. They're not megafauna and charismatic in that way, but they tell a story and they tell us what they are. And we can now take these specimens and use x-rays, and we can build a 3D model of it. So we no longer have to skin it and peel it and put it in a beetle colony to get the skeleton. We can leave the animal intact and see the entirety of the skeleton digitally. We can color the bones by what they are, and then we can create these captivating 3D models either on the screen or that you can print out so that people can see them. And so when you work with something that's really small, being able to blow it up into huge sizes so you can look at it in detail is really fun. And it's really a lot of things we're doing right now.
So is CT scanning for this kind of work a new area for you?
For me, it is, yeah. Like I came from a background of molecular work. Like we would sequence DNA and try and build trees to understand family tree relationships of species. When I came here 9 years ago, I really wanted to make use of this amazing collection. It's the 10th biggest collection of fishes in North America. It's massive.
All right, I think I saw University of Washington has like a walloper of a collection.
They have a huge collection.
Is that just they're coastal, and
Coastal,
so they kind
of
we can't beat them because
they have a lot of larvae. Yeah, yeah.
So also New Mexico.
New Mexico,
interestingly, wasn't it? They had a really huge, like 5 million, or
something like that. Yeah, it's massive.
So the things I've learned in prep for this. So tell us a little bit. So obviously you said you've been here 9 years, and I think were you at the Smithsonian just prior to that?
Yep.
What kinds of things were you doing there, and what are you doing here that's a little bit different?
So there I was a postdoc. So I'd finished my PhD and I was continuing on the research track while looking for a job. And in that project, I was working on a group of South American fishes that are called anastomoids, which includes 4 different lineages. And the people I was working with, Rich Vary was his name. He, over the course of 20 years, had looked at all these different groups and had described the anatomy in detail so that he could code all of the pieces of anatomy as ones or zeros to make this what we call a matrix so that he could analyze it. And he'd done that for each group at a time. And then
what was his goal? What was he trying to
understand the history of the group, to understand the evolutionary history of those lineages. So my job in that place was pulling all of that together into one giant matrix and then filling out that matrix for 280 species for like 500 characters. And so I spent 2 years under a microscope, basically just observing and counting.
This does not sound fun like night at the museum or something like that.
It was fun. It was fun. But it was a lot of work.
All right, so a lot of your work focuses on biodiversity, and it's a word we hear a lot. It's contentious, obviously in some circles. For someone who's not a scientist, how would you explain why biodiversity matters?
It's a great question. I think species and lineages that exist in nature have inherent value just because they've come into being. They have this long trajectory.
That's very spiritual and philosophical.
Yeah.
And,
so they have that inherent value. And the way I look at it is it's my job and people like me's job to try and understand that history. And by describing species or understanding how they're related, we put the context of not only that species or things that they might be closely related to or things that might be distributed in the same places. We put them in the context of the history of not only that group, but of the history of the earth and how things have moved around. And so it sort of just informs us about the world that we live in a way that I find incredibly rewarding and meaningful.
So do we have a sense of how many species there are? I mean, even just in the herps, that's like herpetology is
amphibians and reptiles.
Okay.
Yeah.
And
I think in herps, it's probably better known. I'm the curator of the fishes, amphibians, and reptiles. My background is almost exclusively in fishes, though I have done a couple of projects in snakes. In terms of vertebrate diversity, I'm sort of sidestepping your question to come at it from another angle. Fishes, there are more than about 37,000 described species of fishes on the planet. And as I mentioned a little earlier, we're describing about one a day every year and have been for decades. So that number is continuing to increase. When you add up all of the other vertebrate groups, the amphibians, the reptiles, the birds, and the mammals, those all together don't sum to 37,000. So the fish diversity in vertebrates, fish win, absolutely, hands down.
Well, so I mean, if we, what do we learn by really determining a total number, and how does that inform how we treat wetlands and water management and conservation and those kinds of things?
A lot of times when you can have an isolated area, a pool, a pond, whatever, you can sometimes have something that's unique there. And so when you have those, it gives you a reason to sort of think this is a very special area and we should conserve it. We should think about this in the scheme of all of this, because it's like the devil's hole pupfish. It'll never come back to existence right in the Southwest, but it's in dwindling numbers. So it just sort of gives us a window into how these things come into being. And so, we'll never be able to stop development of places or anything completely, but by understanding where things are, we can get a sense of how to think about those regions as unique in the global history of evolution.
So are there, I think we think of extinctions and losses as kind of a one-way street. Are there success stories in your kind of research or experiments, or even just in kind of what you've seen at the museum? And I actually was reading this morning that I've done a lot of work on the Apalachicola Bay and oystering in Apalachicola Bay, and they just reopened or are in the process of reopening it for wild oystering, which signals that maybe water conditions and that oysters are kind of coming back. So where are some other kind of success stories recently?
Yeah, in vertebrates, there were some droughts last summer or the summer before that in Tennessee, where the Tennessee Aquarium actually went out and got all of these fish from the creek because the creek was going dry. And so they have these fish in aquaria that they're trying to maintain and save so that when the water does come back, they can put them back out in the wild. So they're
basically, they only exist in captivity, like the rhino, white rhino or that kind of thing?
Yep.
Huh. Oh, gosh, that's
it's a lot of pressure.
Precarious.
Yeah, exactly.
Right? Yeah. So how is the health of natural history collections? Are we moving in the right direction? Are we not? I mean, in the world of fish, are we gaining ground or losing?
In terms of what we know about the biodiversity, or in terms of how the
specimen collection?
We in general are contributing far less specimens to natural history museums than we have in the past. We have a paper published a few years ago looking at data. So one of the things that's happened with natural history collections is that we have digitized everything. And so we have these databases and we can push that information out so that the public can see it. And then there are these aggregators that pull all of those different collections together, and you can search globally what we have in terms of the collections. And my colleague Vanya, who's the curator of the birds at the museum, and I and his brother put together a paper where we looked at invertebrates, how many specimens of vertebrates are being added to collections annually. And the trend, you can see doing the spike, and then it's just slowly tapered off. So
is this a citizen scientist kind of problem? Are we not? We're not pulling our weight in terms of collecting
exactly.
Specimens?
Yeah, and there's a thought that maybe we've already got these. Do we need more? But my counter to that is we definitely need more because things are changing environmentally, climatically. We're seeing that happen.
You're saying species themselves are changing and adapting, or
no, they're living through this thing that's happening right now. Yes, species are changing. We see these changes over time, but we are able to, if we don't continue collecting during this time of change, I think it's a disservice to the animals that have been collected in the past that we're using with new technologies. And I like to think that in 100 years from now, who knows what sort of technologies people will bring to bear on questions. And if we don't have things sampled through that whole time, as more and more people are using collections, we have fewer and fewer specimens. So we need to continue adding those to the collections.
So has digitizing allowed for more kind of open source or more sharing or more kind of we're all in this together collections that kind of get integrated across museums?
Yeah. The National Science Foundation has this had this program they called thematic collection networks. And when I first got here, we had a grant. There were 18 or 19 museums in the U.S. that had this grant called OVERT, which was the Open Exploration of Vertebrate Diversity. And it was the idea was to CT scan every genus of vertebrates and make that data publicly accessible, similar to the way that GenBank is an accessible database for DNA sequence data. When you sequence DNA and you publish a paper, most journals require you to put that data on this public warehouse so anybody can go look at it. Anatomy has been historically sort of isolating. People just sort of work on their own thing and then they publish it. But we were trying to push this idea that it should be open source. And so we have this website that was created before we started OVERT, but it's out there that hosts all this 3D CT data called Morphosource. And it's a way of allowing people access to these data that are sometimes locked behind museum doors.
Oh, that sounds incredibly exciting. So I want to bring it around to kind of the optimism, and that seems
Yeah.
like a piece of it right there.
Yeah.
Just the new technology's ability to share data like this, and maybe in some ways to preserve data kind of in perpetuity. What are the other things that kind of give you or are exciting to you right now?
Yeah. One of the things that's really exciting is thinking about how many species are still out there, like that we don't have a name for. And by building these networks of museums and working together, we can really sort of start to answer some of those questions. And there's a whole lot of excitement in my mind around species descriptions. It's not like the most exciting.
But we don't name them after ourselves anymore, right? So there's not going to be the Dillman fish anytime soon?
I think technically by the rules of nomenclature, as esoteric as that sounds, you can't name something after yourself. Mm-hmm. Somebody else would have to name it after you. But your friend
could name it after you.
Yeah.
Okay.
There is a slight push, I think, in the U.S. to stop that. But I think that's a little sort of, it's not up to the U.S. to decide in that way what other people in countries are doing.
Sure.
And I think we've had this historical sort of heavy hand in that way of like, oh, it was fine when we did it, but now nobody else should do it, is the way I take it. And I think that as people are, especially like through the global south, as more species are being described, they should be able to describe and name things however they would like. And it shouldn't be a dictation from somewhere else.
Yeah, sure. So for people who are listening, who might be interested in this topic, kind of where do they start? What's a way to get involved, learn more?
Yeah, it's a great question. Like I think, so the Museum of Vertebrates at Cornell is a little different than like a Smithsonian or American Museum in that we have this major research collection, but we don't really have a public space. There's a small exhibit space at the Lab of Ornithology, focuses heavily on birds. Very well done, very exciting. But you don't go to this museum and see animals in
We can't see
display.
the 1.3 million.
No, you can. You just have to, tour it with a group or make reservations or contact us. Reservation sounds like we have a formal thing, but you just let us know and we can show people around. But to get involved with it, I think it just requires like reaching out to people. Like, if you're really passionate about something like I am with fishes, like my earliest memory is being in streams when I was like 3. That's my earliest memory. So I've always been drawn to it. And I think what you do is you just reach out to people and you talk about it and you go to open house events and you interact. And we end up finding people that want to come in and volunteer and do things. And
well, it does feel like a great community to be in.
Yeah.
If you're interested, birds, fish,
fish, amphibians,
think,
reptiles. Yeah. And it's a supportive group for the most part of people. And it's a lot of fun when people are excited by what you do, it's natural to want to bring them in and have them sort of help out.
So how about for kids? So I think kids are obviously kind of, I don't know, it seems to me like kids go through the dinosaur phase and then into the kind of the
fish, not everybody can do that. Yeah.
Frogs, et cetera.
Absolutely. Yeah, I think really, again, it's a matter of like when these open house events happen, make contact with people. I think reading a lot of material online is becoming more and more available. You can spend time just at the library going through books and sort of getting into, and I get letters from middle schoolers saying, I'm really interested in this. I would love to set up a time to chat. And so it's just, I think being proactive is really what it is. And then as you progress towards college, finding a school and a group of people who are like-minded. Cornell is really unique, I think, in a lot of ways, because we have these courses called the ologies, which are like herpetology, ichthyology, mammalogy, ornithology, so birds, mammals, amphibians, reptiles, fishes. And those courses have come and gone at some places, but Cornell has always had them and has always taught them. And we end up with a large number of undergraduate students that come here specifically because they're focused on understanding biodiversity through the ologies. And they end up finding their way out to the museum. And we have this really wonderful group of undergrads that spends time out there working in the collections, conducting research, and otherwise building this sort of cohesive group of people that are just passionate about biodiversity.
Wonderful. So I want to steal a page from Ezra Klein, who always asks his guests about book recommendations, and he asks for 3. I'm only going to ask you for 1.
Okay.
So what should people be reading if they're interested in Fish and herps.
Fish and herp. I actually will go a little more broadly, and I'll say Richard Conniff has a book called The Species Seekers, and it's about, I can't remember the subtitle of it. It's like fools and the mad pursuit of life on Earth. Basically, people who have gone searching for new species and the history of that and where it's at. And it's a fun read. It's a lot of just the sort of the history of this science of understanding biodiversity.
Oh, yeah. Wonderful. That sounds great. All right. So I did look up a bunch of your papers. And so it looks like you've done a lot of work on black bass, Atlantic sturgeon, electric eels. I saw some, rainbow trout, bony tongue fish. What are your favorite fish that you've done work with?
My favorite fish? That's a great
Not
question.
Not to eat, but just to study. Although maybe they go hand in hand. I don't know.
Yeah. The black basses and sunfishes are a cool group. I really like them. I like North American freshwater fishes are really just a really interesting group in my mind. Most of it is endemic, meaning it occurs only in North America. And given our latitude north, across that entire latitude from Europe and Asia, like we have over like 1,000 species and there's several hundred there. So we have this really smaller landmass with a really diverse fauna. And so thinking about how that comes into being is really exciting, and understanding how Earth history is shaped, where things are distributed. And I just think that it's really a fun way to spend one's time.
Great.
Yeah.
Well, Dr. Casey Dillman, thank you so much.